All right, guys, I want to talk to you about some test taking tips for your AP Biology exam. Most of you guys are really well prepared. I want to make sure that you do as well as you possibly can, or as good as you possibly can. The first part of the test is multiple choice and math questions. You've got an hour and a half to answer together 63 multiple choice questions and then 6 grid-in math questions. The math questions are usually pretty easy. So I would recommend that you flip back and do those first. Make sure you get those points. Don't waste a lot of time if you happen to hit one that you don't know. But get the ones that you do know how to do, bubble those in, and then go back and work on the multiple choice. And again, I think you'll find most of those easy. Some though are really long, and one question may take up a whole page in the booklet. If you're running very low on time, I would skip those long single questions. Make sure you get as many answers as you can. Make sure that you wear a watch. Keep up with your time. If you see that there's just no way that you can get to some questions, make sure that you at least bubble in an answer. And what I would recommend is you bubble in the same answer on each one of those. I would say do B. For a long time, people taught C, and the test makers sort of know that. So sometimes they tend to avoid using C as a common answer. So again, if you just run out of time completely and you don't have any guess on something, use B. If you do have time, though, take your time, eliminate all the possible choices that aren't right, and then guess from the ones that are left. There is no guessing penalty, so make sure that every question on your test has an answer bubbled in. Section 2 of your test is your free response section. There's 8 total questions. 2 of them are long free response questions. You may hit a part or a couple of parts on each one of those that you don't know a whole lot about. But there are also going to be parts that you do know a lot about. The key to scoring well on this AP Biology exam is putting what you know on paper. Put every single detail. The assumption of the reader is that you don't know the answer. Prove to them that you do. Put as much on paper as you can. A long free response, a good answer for one of those should take probably a whole page or near a whole page. I would recommend you do them a section at a time. Usually they're going to be A, B, C, D. Write down A, answer part A. Write down B, answer part B. Write down C, and so on. Your answers do have to be in complete sentences. Don't use list or something like that. You can use a picture, but to get points, you must explain your picture. And also for you guys especially, make sure that you write neatly, and I would recommend that you print. There's also 6 short free response questions. The ones about all of those, I think a good thorough paragraph, 5 to 6 sentence paragraph, if you're direct and to the point, should be plenty to answer those questions. But again, make sure you put down everything you know and get all the possible points. To do this free response section, you're going to have an hour and 20 minutes to write. Plus, before you start, you're going to have a 10-minute period where they give you the questions and you have an opportunity to make outlines of what you're going to write. Use that time wisely. Write down key words and key phrases that you want to use in your essay. And then when you get the go-ahead to start your hour and 20 minutes, write all those things down. Doing well on the free response is the key to doing well on this test. Put everything you know down on paper. It's important to know that you can begin to write your actual free response answers during the 10-minute planning or outlining period. But you want to make sure to go ahead and outline and plan before you start to write those answers. Write on the book, write on the questions, make a list of important topics or concepts that you need to include in each answer. Use that planning time wisely, but when you finish planning, you can begin to go ahead and write before the 10-minute planning or outlining period is over. It's also very important to pay attention to all of the bold verbs in the FRQ questions. Do what those verbs tell you to do. If you do those things, you will earn points. The points come by addressing the verbs in the question. You're going to be asked to justify your answer on almost every biology FRQ question. To do that, use evidence and/or data and make sure that you support your conclusion. Use complete sentences. Describe everything that you know that will justify or back up the conclusion that you reached. That being said, I want to do a couple of review points, and I'm going to start with evolution. I'm going to do a little bit with plants, and then finally a couple of genetics terms that I want to make sure you know. Remember, this is the first video in a series of 3. Please take the time to watch all of them on the weekend before the test. This evolution stuff, I think you guys are pretty well prepared on, but make sure you know the definition. Evolution is a change in the allele frequency of a population over time. Individuals cannot evolve. Groups evolve. The population is the biological unit of evolution. Another thing that I'll mention about evolution is that you can't pass on traits that you acquire during your lifetime. A guy named Lamarck had a theory of evolution based on the idea that acquired characteristics could be passed on to your offspring. This was the example we talked about of like a giraffe stretching its neck, and then its babies would then be born with a longer neck. Make sure you know that that doesn't make sense. You can't pass on acquired characteristics. Another thing that we want to mention is macroevolution, or sometimes it's called speciation. For one species, for one population of a species to evolve into separate species, it can only happen if you have two or more populations that are genetically isolated. That means they're separated somehow from each other, and that they're not sharing their genes. There's no gene flow between the two groups. That gene flow is cut off. And because they're separated, and because they're isolated from each other and gene flow is cut off, mutations accumulate in those two groups. And since mutations are random events, the mutations that happen in those two groups are different. So given enough separation and enough time, those groups may evolve into separate species. There's 2 patterns of speciation that I want to make sure you know really well. The first one is allopatric, and that word literally means different fatherlands or different places. This is when you have 2 populations of the same species that are separated by some kind of geographical barrier. It could be a river, it could just be distance, it could be a mountain. But because they're separated, and because there's no gene flow between those 2 populations, given enough time and enough mutations, they evolve into separate species. The second example is sympatric speciation. This word literally means same place. This is where you have a group of organisms of the same species living in the same place at the same time, but they're still separated somehow. It can be that they have different mating times. Maybe they have some kind of temporal separation. Their breeding times, or if you're a plant, your flowering times are different. So there's no gene flow between those that flower or breed at different times. It can also be due to polyploidy. And this is a plant thing. It doesn't usually work in animals. And this is where, through some strange breeding patterns, a plant gets extra chromosomes. And therefore it can't breed with the plants that it came from. That's called polyploidy. 2 examples of sympatric speciation: temporal separation because of different breeding times, or separations due to chromosome number, polyploidy. All right, we also want to talk about once you have 2 separate species, how do species continue to be separate? The most common definition of a species is 2 groups of similar organisms, or again, let me start over on that. A species is a group of similar organisms that can interbreed with each other and that produce fertile offspring. But let's say we have 2 separate species, but maybe closely related. There are these things called reproductive oscillating mechanisms that keep them as separate species. Now, they can be prezygotic, which means they prevent the 2 organisms from actually making a zygote, a baby. Those prezygotic oscillating mechanisms can be temporal, meaning that they have different breeding times probably, so that they can't mate because they breed at different times. It can be mechanical, that just their reproductive organs, they don't fit, they don't work together. It can be gametic, that maybe they do mate, and their sperm and eggs get to each other. But when they get to each other, they don't work together. They can't produce a viable zygote. Another prezygotic mechanism can be habitat oscillation, that they just live in different places and they're not around each other. And then finally, can also be behavioral, that they have different maybe mating attraction behaviors, like a bird song or a fly pattern or a mating dance. And because they don't recognize the mating behaviors, they don't tend to mate with each other. There are also oscillating mechanisms that are called postzygotic, meaning that maybe the two groups are very much alike, and that given the opportunity, they'll breed, and they can actually make a zygote. Oftentimes, though, those zygotes, those mixed babies, the hybrids, are inviable, meaning they don't have the ability to live. They're inviable. It can also be like the situation with a mule, that the mule is born and it's very strong, but that it's sterile, so it can't make babies of its own. And then finally, we also get oftentimes in plants what's called hybrid breakdown, where a hybrid is produced, but after several generations, it either loses its ability to live or to reproduce. Again, these are reproductive oscillating mechanisms, things that keep one species separate from another. I also want to briefly mention some patterns or types of evolution. And these are fairly simple to understand, but the terminology is something you need to be able to know and explain. One pattern of evolution is convergent. You guys should know by now that converge means to come together. This is when two organisms that aren't closely related through evolution evolve to look very similar because they're living in very similar habitats, environments. Good examples of this would be like the shark and the dolphin, a fish and a mammal, or the sugar glider, which is a marsupial living over in Australia, and a flying squirrel, which is a placental mammal living in North America. Even though they're on different continents, the places that they live and the habitats they live in, the niches they occupy are very similar. So they've evolved to look very similar because that's what it takes to succeed in that particular niche or environment. The more common type of evolution that we talk about is divergent. And you guys know that diverge means to go apart, to become different. Divergent evolution is when you have a group of the same species of organism, you put populations in different places or different niches, and they evolve to become different species. A good example of divergent evolution is adaptive radiation. This is a key term to know. This is when you take a population of organisms of the same species, you put them into a new environment. And because of things like competition, they begin to evolve very quickly into different species. They begin to speciate. And they're doing that in order to avoid competition, because there's some kind of limited resources like food or space. And it also allows them to move into new unoccupied niches in that particular environment. Some good examples of this would be Darwin's finches when they were placed onto the Galapagos Islands, and also the mammals that were on Earth right after the extinction of dinosaurs. There were all kinds of new niches available, and they evolved to fill them. They speciated to fill them. Another important type of evolution we want to talk about is coevolution. And this is when two different species of organisms evolve together. They help to shape each other's appearance, behavior, eating habits. A good example, or two good examples of coevolution are first of all, plants and their pollinators. Plants have evolved to have a specific color, a specific smell, a specific shape. And a lot of that has happened to attract and to lure in their pollinators. And on the flip side of that, the pollinators have a specific shaped beak or eating apparatus to fit that particular plant that they tend to visit. So that's a good example of coevolution. Another one would be like parasites and their host. Parasites have evolved different ways to get into the host, different ways to avoid the immune system. It's sort of like an arms race between the parasite and the host. We might also add up here predators and prey. They co-evolve together as well. If we happen to mention the word microevolution, that means there's small changes in a population, but not enough to cause speciation to happen. A good example of microevolution might be the peppered moth example that we talked about during the Industrial Revolution, or the emergence of antibiotic resistance in bacteria or pesticide resistance in different kinds of insects. It's small changes in a population, but not enough to cause them to become a new species. And then the last evolution.
Hardy and Weinberg were two scientists who looked at the idea of genetic equilibrium. Genetic equilibrium is an idea or a situation, a theoretical situation, when allele frequencies in the population are not changing. Today, we also refer to genetic equilibrium as Hardy-Weinberg equilibrium. Again, Hardy-Weinberg equilibrium is essentially a theoretical condition when evolution is not happening. Probably doesn't completely exist in the real world. According to Hardy and Weinberg, there are 5 requirements that must be in place for a population to enter into and remain in genetic equilibrium. Those 5 requirements are: number 1, there can be no mutations in the population. In particular, we're talking about here germ cell mutations, because germ cells are the cells that end up going through meiosis and becoming gametes. The genes in those cells are the only ones that are passed on. Somatic cell mutations don't matter so much here. Somatic cell mutations, or somatic cells rather, are just body cells, and the genes in those cells are not passed on to the offspring. Number 2, there can be no immigration, either coming into the population or exiting the population. Sometimes we might refer to that as gene flow. When immigration happens, new alleles might be introduced into the population, or some of those alleles, unique alleles, might leave the population. 3, the population must be really large so that we don't get genetic drift. Genetic drift is an unpredicted change in allele frequency due to chance. It almost always occurs in a small and usually an isolated population. 4, there must be no natural selection. And 5, there must be no sexual selection. Mating must be random. Mates aren't chosen based on some characteristic in this case. So again, Hardy and Weinberg equilibrium is a theoretical situation in which evolution is not happening, in which allele frequencies are not changing. So essentially, by defining this theoretical case, Hardy and Weinberg were able to define the things that cause evolution. If you look at the negative of the 5 requirements for Hardy-Weinberg evolution, you're really looking at the 5 things that might contribute to causing evolution. Mutations can cause evolution. Immigration can cause evolution. Genetic drift can cause evolution. Natural selection can cause evolution. And non-random mating or sexual selection can also contribute to evolution. Now you can see at the bottom left of your screen, down here, a section off the AP Biology formula sheet. These are the Hardy-Weinberg equations. Hardy and Weinberg developed a system for using these equations for calculating allele frequencies. In these equations, P represents the frequency of the dominant allele in the population. Q represents the frequency of the recessive allele in the population. And notice that those 2 frequencies must add together to be 1. We might think of it in terms of 100%. When we're using these equations, we want to work in decimal. So if you have a percentage, you want to convert that percentage to decimal and use decimal in these equations. The top Hardy-Weinberg equation deals with genotype frequencies. Remember, genotypes are the actual gene combinations an organism possesses. So P squared in this top equation, P squared represents the frequency of homozygous dominant individuals, individuals who have 2 copies of the dominant allele. 2PQ represents organisms that are heterozygous, organisms that have 1 dominant and 1 recessive allele for a particular condition. And finally, Q square represents individuals that are homozygous and recessive, individuals that have 2 copies of the recessive allele for the condition that we happen to be looking for. It's important to realize that the Hardy-Weinberg equations can only be used if the population is in genetic equilibrium. If the problem stem doesn't state that the population is in Hardy-Weinberg or genetic equilibrium, you need to assume that it isn't. If the population isn't in genetic equilibrium, you must count the alleles in order to determine the allele frequencies. We're going to see problems in the coming problems that show you both approaches to calculating allele frequencies. We'll start with some problems at which the population is at genetic equilibrium. And we'll end this presentation with some problems in which the population isn't in genetic equilibrium. Make sure that you can work both types of problems. And the rest of this video is to look at some example problems we're going to try to look at pretty much every possible situation that AP folks might throw at you in terms of Hardy-Weinberg problems. So the first one's a very simple one. It says, in humans, brown eyes are dominant over blue eyes. And that's an oversimplification, but let's go with it for the problem. In a population of 1,000 individuals, 750 have brown eyes. Assume the population is in Hardy-Weinberg equilibrium. Part A, what are the frequencies of the dominant and recessive alleles? Part B, how many individuals would you expect to be heterozygous? And Part C, what is the expected frequency of each possible genotype? So, what they gave us in this problem is that we had 750 individuals with brown eyes. So 750 with brown eyes. They told us we had 1,000 individuals. So if 750 had brown, that means that 250 have blue. Now the thing that we really have to look at here, and what you typically want to find in a Hardy-Weinberg problem first, when possible, is the percentage or the frequency of homozygous dominant individuals. Let me say that again, homozygous recessive individuals. Because notice in this equation, p squared and 2pq individuals, it's very hard to tell those guys apart. By appearance. So again, we had 750 that were brown. Individuals that are p squared and 2pq, they both have a dominant allele, which means they're both brown. Whereas q squared individuals, they have 2 blue alleles. That's why they're blue. So in this case, we have 250 individuals with blue eyes out of a total of 1,000 individuals. That's our frequency of homozygous recessive individuals, blue-eyed individuals. So let's divide that out. 250 divided by 1,000, and that gives us 0.25. That is q squared. That is our frequency of individuals that are homozygous recessive. Well, the next thing that we want to do is to go ahead and just do what the math says. Let's take the square root of q squared. So if we go 0.25, and we take the square root of 0.25, well, the square root of q squared is q. And the square root of 0.25 is 0.5. So that gives us q. Q is the frequency of the recessive allele. And if we look back at our problem, that's one of the first things that it asks us to find. Part A says, what is the frequency of the dominant and the recessive allele? Q is the frequency of the recessive allele. If we look back at this equation right here, we also find that P plus Q equals 1. Remember that P is the frequency of the dominant allele. So we already know the recessive allele is 0.5. P and Q together have to add up to be 1. So if Q is 0.5, P also has to equal 0.5. So our answers for part A, frequency of the dominant allele, P, 0.5. Frequency of the recessive allele, Q, also 0.5. Now if we look at part B, how many individuals would you expect to be heterozygous? So remember, in our top Hardy-Weinberg equation, this one up top up here, 2pq, those are the individuals that are heterozygous. So again, the 2pq individuals are the heterozygotes. 2pq. So to calculate the frequency of heterozygous individuals, we're going to just do what the math says. We're going to say 2 times p times q. So 2 times p, which we just found to be 0.5, times q, which we also found to be 0.5. We multiply those together, 2 times 0.5 times 0.5, and that gives us 0.5. Now that 0.5 is the frequency of heterozygous individuals. 50% of this population is expected to be heterozygous. The question here, though, didn't ask us for the frequency or the percentage of heterozygotes. It asked us for how many heterozygous individuals we expected. So we expect 50% of a total population size of 1,000. So we're going to take 0.50 times 1,000, and that tells us that we're expecting 500 heterozygous individuals. Part C, the last one, wants to know the expected frequency of each possible genotype. Remember that our top equation, this one up here, is the genotype equation. So we're looking for the frequency of homozygous dominant, the frequency of heterozygous, and the frequency of homozygous recessive individuals. We've already found two of those. We found up here that the frequency of homozygous recessive individuals, Q squared, was 0.25. So let's write that down. We also, here in the middle section, found that the frequency of heterozygous individuals, 2pq, was 0.50. All right, what we have left is the frequency of homozygous dominant individuals, p squared. To find that, we just simply need to square p. Remember, we found that p, the frequency of the dominant allele, is 0.5. So we need to square 0.5. .5 squared is 0.25. So our answer to part C is the frequency of homozygous dominant individuals, p squared, 0.25. The frequency of heterozygous individuals, 2pq, is 0.50. And the frequency of homozygous recessive individuals, q squared, is 0.25. All right, here's problem 3. In a given population of mice, brown hair, big B, is dominant to blonde hair, little b. Within the population of mice, there are 125 brown-haired mice out of a population of 300. Assume the population is in Hardy-Weinberg equilibrium. A, what are the frequencies of the dominant and recessive alleles? That's P and Q. B, what is the predicted frequency of heterozygous mice? C, the predicted frequency of homozygous brown mice. And D, how many mice are heterozygous? So let's take a look and see if we can't work this problem out. So the first question wants us to find the frequency of dominant and recessive alleles. The problem told us that there were 125 mice that were brown. We'll just use BR. There were 300 total mice. So if there are 125 that are brown, that should tell us there's also 175 that are blonde. Those have to add up to be 300. Remember, whenever possible, we want to find the frequency of homozygous recessive individuals. That's Q squared. If we can find Q squared, we can find all the other variables. In this case, Q squared is going to be represented by the blind mice, because to be blind, since it's a recessive trait, you have to be homozygous for that condition. So we're going to take 175 out of 300, and that fraction equals Q squared. Our frequency of individuals who have two blind alleles. They're homozygous recessive for blind. So if we divide out that fraction, 175 divided by 300, let's go ahead and put that in decimal form. That gives us 0.583. Now, the first thing we're trying to find here are the frequencies of the dominant and recessive alleles. So to find the frequency of the recessive allele Q, we need to take the square root of Q squared. So if we take the square root of 0.583, when we do that, we get Q equals 0.764. So that's the frequency of the recessive allele. We also know that P plus Q equals 1 from our top left equation there. So if Q is 0.764, P must equal 1 minus 0.764. So P is equal to 0.236. So we've now answered part A. We have the frequency of the dominant allele, 0.236, and the frequency of the recessive allele, 0.764. Now part B asks us the predicted frequency of heterozygous mice. Remember that in this top equation, heterozygotes are represented by 2pq. So we're simply going to do what the math tells us to do. We're going to take 2 times p, which we just found to be 0.236, times q, which we just found to be 0.764. And now we multiply that out. 2 times 0.236 times 0.746, and we end up with 0.352. That's our frequency of heterozygous individuals. We're saying that about 35.2% of the individuals in this population should be heterozygous. Part C asks us for the frequency of homozygous brown mice. Now remember that brown is dominant. So homozygous brown mice are going to be homozygous dominant, or p squared. So again, we're going to do what the math tells us to do. We're going to take p, which we found to be 0.236, and we're simply going to square that. 0.236 squared gives us 0.056. So we're saying about 5.6% of this population should be homozygous dominant for brown. Alright, that leaves us with only part D. And in this case, we want to know how many mice are expected to be heterozygous. Here, we found the expected frequency of heterozygous mice. But what we want to find now is the number of mice. To do that, we're going to take the frequency, 0.352, and we're going to multiply it by the size of our population, which in this case we had 300 total mice. So 0.352, the frequency of heterozygotes, times 300, the total population size, that results in a total expected number of heterozygous mice of 105.6. All right, let's move on to the next problem. This problem, like problem 2, gives us information about alleles instead of about genotypes or phenotypes. So we have to approach working this problem in a little different way than we did problems 1 and 3. Let's look at the problem first. In a given population of 200 insects, there exist 121 alleles for the recessive trait of purple exoskeleton color. Assume the population is in Hardy-Weinberg equilibrium. Part A, what are the frequencies of the dominant and recessive alleles, P and Q? Part B, what is the expected number of insects with purple exoskeletons? And part C, give the 3 genotypic frequencies for this population. All right, so first of all, we're given that there's a total population of 200 insects. Just like we did in problem number 2, we need to remember that each individual typically has 2 alleles for each trait. So we're going to take our total number of insects, 200, we're going to multiply that by 2 alleles per insect. That's going to tell us that for this trait there's about 400 total alleles in the gene pool. Now the stem of the question told us that out of that 400, about 121 of those, out of our total of 400, were recessive purple alleles. That should tell us, or that should represent for us Q, because remember that Q is the frequency of the recessive allele. We have 121 recessive alleles out of a total of 400. So let's divide that out, put it in decimal form. So 121 divided by 400 gives us, for Q, 0.303. So we now have a frequency of a recessive allele. Now again, go back to this equation over here, P plus Q equals 1. To find P, we're simply going to go 1 minus Q. So if we go 1 minus 0.303, That gives us a frequency of the dominant allele P to be 0.697. So we've now answered part A. 69.7% of the alleles are dominant alleles and 30.3% are recessive alleles. Part B wanted to know what is the expected number of insects with purple exoskeletons. Remember, the purple is recessive, so the only way to exhibit a recessive phenotype is to be homozygous recessive. And in our top left equation, homozygous recessive individuals are represented as Q squared. These are going to be the only purple insects. So we need to do what the math says. Take Q and square Q. So in this case, that's going to be 0.303 squared. And when we square our 0.303, we end up with a frequency of homozygous recessive individuals of 0.092. Now some people would stop and probably write that down as the answer. This question doesn't ask for the frequency of purple exoskeleton insects. It asks for the number of them. So to find the number, we need to take the frequency, 0.092, and we need to multiply that by the total size of our population, which in this case, the stem of the problem told us was 200. So 0.092 times 200, we end up with an expected number of purple of insects with purple exoskeletons to be about 18.4. So that's our answer to part B. Final thing we wanted to find were the genotypic frequencies. Remember that this equation up here is our genotype equation. We've already answered part of this equation. We're trying to find the frequency of homozygous dominant individuals, heterozygous individuals, and homozygous recessive individuals. Right here, we found the frequency of homozygous recessive individuals, Q squared. So Q squared came out to be 0.092. All right, again, Q squared, our frequency of homozygous recessive individuals, 0.092. All right, now let's find the frequency of heterozygotes, 2pq. So to find that, we're simply going to multiply 2 times p, which was 0.697, times q, which was 0.303. When we do that, 2 times 0.697 times 0.303, we end up with an expected frequency of heterozygous individuals to be 0.422. And then finally, we need to find the frequency of homozygous dominant individuals. Those are p squared. So we need to take p, 0.697, and simply square it. And when we do that, we end up with 0.486. So our answer for part C, our frequency of homozygous dominant individuals, 0.486. Our frequency of heterozygous individuals, 0.422, that's 2pq. And our frequency of homozygous recessive individuals, 0.092. Now one wrinkle they might throw at you, instead of asking for genotypic frequencies, they might ask for phenotypic frequencies. What you've got to realize is that in a problem like this, even though p squared individuals and 2pq individuals have different genotypes, since they both have a dominant allele, their phenotypes look the same. So to calculate the phenotypic frequency, you would add together 0.422 and 0.486. So let's see, that would give you 0.980. Those individuals all look the same. So they're not going to have purple exoskeleton color. Let's say the other color was green. So we're saying about 90.8% of this population would have green exoskeleton colors, while 0.092 would have purple exoskeleton colors. So the phenotypic frequencies would be 0.908 green and 0.092 purple. All right, we've got two.
All right, so here's an example problem for finding allele frequencies where the population is not in genetic equilibrium. And remember, if that is true, you cannot use the Hardy-Weinberg equations. So let's look at the problem. Out of a population of 60 people in a small rural town in Kentucky, one person has blue skin. It's an autosomal recessive trait. 3 people in the population are carriers for the trait. How many total alleles, blue and normal, are in the gene pool for this trait? And again, the population is not in Hardy-Weinberg equilibrium, and we're going to give our answer as a whole number. Well, many human traits are controlled by 2 alleles, one from each parent. We're going to assume that's true here. So we have 60 people. Each of those have 2 alleles each for this particular trait. And that means for this trait, there are 120 alleles in our gene pool. And that's our answer for part A. Now for part B, we want to find the allelic frequencies or allele frequencies for this trait. That's asking us to find P and Q. And again, we can't use the Hardy-Weinberg equations because our population is not in genetic equilibrium. So we're going to have to count alleles. We're going to give our final answer as a decimal rounded to the thousandths place. What we've got to realize here, though, is that, first of all, this is an autosomal recessive trait. One person has the trait. That means that person has to be homozygous dominant. That person has to have 2 alleles, 2 blue alleles, 2 recessive blue alleles. There are also 3 people who are carriers for the trait. That means they have 1 blue recessive allele each. So that's plus 3 more blue alleles. That means we have 5 total blue alleles. 5 blue alleles, 5 recessive alleles out of a total of 120. That fraction is going to be equal to Q, the frequency of the recessive allele. So if we go 5 divided by 120, that gives us 0.042. Now, if 5 out of 120 alleles are recessive, that means 115 out of 20 are dominant. That fraction is going to be equal to P. So if we divide 115 by 120, we end up with a P in decimal form, and that's 0.958. So those answers, Q equals 0.042 and P equals 0.958. Those are our answers to part B. Those are the allele frequencies. Now let's go down and look at part C. If the population now enters genetic equilibrium, which means we can use the Hardy-Weinberg equations now, and produces 140 children, how many of these children will be carriers for blue skin? So if the population is in genetic equilibrium, that means the allele frequencies are not changing from generation to generation. So these numbers were our allele frequencies in our first generation. They're also going to be our allele frequencies in the generation asked about in part C. We're looking for the people who are carriers. Carriers means that they're heterozygous. And we know in our Hardy-Weinberg equation, this one, these individuals, the 2PQ individuals, are heterozygotes, or in this case, are carriers. So to calculate how many of the children are going to be carriers, we're going to go 2 times P times Q. So in this case, 2 times 0.958 times 0.042, and we end up with 2PQ equal to 0.080. Now that's the frequency of heterozygotes. That's not the answer to this question. So we need to go just a little bit further to get the answer here. So essentially, this is 8% of the population is heterozygous. So we're going to take the frequency of heterozygotes, 0.08. We're going to multiply that by the population of the generation we're looking at, 140. So 140 times 0.080. And this is telling us that we should have around 11.2 children who are carriers. And notice that we're rounding our answer to the 10th place. So this is the correct answer for part C. Let's look at one more example problem where a population is not in genetic equilibrium, and that therefore you need to find P and Q using means other than the Hardy-Weinberg equations. So here we go. We have the genetic makeup of a mouse population is provided in the table above. At the start of an experiment, researchers remove all gray mice from the population, resulting in a population that is not in Hardy-Weinberg equilibrium. Calculate the frequency of the recessive allele in the remaining population of mice. Give your answer as a decimal to 2 places. So here's the population after the gray mice are removed. Notice there are no grays. This population is not in Hardy-Weinberg equilibrium, so we can't use the Hardy-Weinberg equations. But what we do know is that there are 1,218 individuals who are carriers and 841 who are homozygous dominant. So let's just use those carriers for a second. 1,218 carriers, they have 1 dominant allele each and 1 recessive allele each. That means that we have 1,218 total recessive alleles. Well, if you note, we have 2,059 mice. Each one of those mice have 2 alleles for this trait each. So if we go 2059 times 2, our total number of alleles for this population are 4,118. So the fraction 1218 out of 4,118, that is Q, the frequency of our recessive allele. So let's divide that out. 1218 divided by 4118. That gives us Q equals to 0.2957. So we're going to run that to 0.30. All right, now on the other hand, to find P, we could just look and see that P has to be 70%. But the other way to look at it would be these individuals, these 841, they have 2 dominant alleles apiece. So 841 times 2, that's 1,682. These individuals, the heterozygotes, have 1 dominant allele each, so 1,218. So if we add 1,682 plus 1,218, that gives us a total of 2,900 dominant alleles. 2,900 dominant alleles out of 4,118 total alleles. That fraction is P. So let's go 2,900 divided by 4118, and that ends up giving us 0.70 as P. So moral of the story, if the population is not in genetic equilibrium, you're going to have to count the alleles. You cannot use the Hardy-Weinberg equations. You can only use those when the problem states that the population is in genetic equilibrium or Hardy-Weinberg equilibrium. Hope that helps. Additional topic that we want to cover about evolution. We want to talk about the evidence that scientists have for evolution. An important word that will show up on the AP exam over and over again is phylogeny, or you might hear it said as phylogenetics. This is essentially the evolutionary history of a species, the pattern in which different species evolved from common ancestors. So some proof that scientists have for evolution, number one, are homologous features, or we might call them homologies. These are structures in different species that are similar because of common ancestry. Think of it like the arm of a human, the wing of a bat, the flipper of a whale. They all have very similar bone structure. They all look very similar because there's common DNA from a common ancestor that caused them to be the way that they are. When we talk about the ultimate homology, the thing that proves the most that organisms are related, it's shared DNA or RNA or protein structure. Species share similar DNA sequences because they shared common ancestors. The more two species are separated over time, the more time there's been for mutations to build up, so the more different they will be. The more closely they are in terms of development through evolutionary time, the less mutations would have occurred, and therefore the more similar their DNA sequences are. So if you're asked to show that two organisms are related, that they've evolved from a common ancestor, the most important thing for you to say is to sequence their DNA for a common gene or a set of common genes and compare those DNA sequences. We might also talk about vestigial organs or vestigial structures. These are structures that are remnants of a structure that was important in evolutionary ancestors. They probably don't have a function anymore in the current ancestor, but they did in the past. For example, the appendix in humans. In our evolutionary ancestors, it's thought that the appendix might have been a structure to help digest cellulose. Reptiles, like snakes, for example, whales, dolphins, they have a vestigial pelvis and vestigial leg bones. And this is to show that they probably evolved from four-legged land-based animals. Snakes probably came from lizards. Whales probably came from some hippopotamus-like organism. Another type of evidence that scientists use is comparative embryology. So here they compare the embryos and how they develop from related species. Let's look at an example of that. So here's some embryos from related vertebrate organisms. We've got a lizard, a tortoise, a pig, and a human. And we noticed in what might be like the first trimester of the pregnancy, it's almost impossible to differentiate those four from each other. They're very, very similar because they all share similar DNA, similar patterns for constructing a new organism. As the pregnancy progresses, notice the organisms become more different. These two, though, still look very much alike, and these two still look very much alike. That's because these two are more related to each other than they are to these two. These two are mammals. These two, over here on the left, are reptiles. So they share even more DNA with each other than they share with the mammals. The mammals share even more DNA with each other than they share with their reptiles. And notice that by the time we get to birth, the two mammals still look lots alike. The two reptiles still look a lot alike. So by comparing embryos, essentially scientists are saying the more alike the embryonic development patterns are, the more related the organisms are in terms of evolution. Another important piece of evidence that scientists use for evolution is comparative biochemistry or comparative molecular biology. We sort of talked about this in the first piece of evidence. This is where you compare the DNA, the RNA, or maybe the protein structures or amino acid sequences of related organisms to see how related they are. You might also look at their metabolic pathways. Got a couple of examples I want to share with you. So here we're looking at the amino acid sequence in hemoglobin. Hemoglobin was chosen because essentially all vertebrates have it. It's an important protein that they have to have. It's a shared set of genes. So we're comparing these different organisms to human hemoglobin. So human to human, there's zero differences between the amino acid sequences. Humans to rhesus monkeys, out of 146 amino acids in the hemoglobin chain, only 8 are different. With mice, 27 of the 146 are different. With chickens, 45 out of the 146 are different. With frogs, 67 out of the 146 are different. And with lampreys, a type of jawless fish, very primitive organism, 125 out of 146 are different. So scientists would argue that the more similar the amino acid sequences or DNA sequences, the more related the organisms are. So humans and rhesus monkeys are closely related. Humans and mice are a little more distantly related. Chickens are even a little bit more distant from humans. Frogs more distant. Lampreys more distant. The pattern here follows the fossil pattern. If we go to the deepest levels of the fossil record where we find vertebrates, the only vertebrates we find down there are fish. And for layer after layer, we only see fish. But we get to some pattern where amphibians evolved and they showed up. So we have a set of rock where there are fish and amphibians for long periods of time. Then along the way, reptiles evolved. So then in the fossil record, we find fish, amphibians, and reptiles. and it stays that way for a long period of time. And then we see fish, amphibians, reptiles, birds, and it's somewhere maybe a little bit later, a little bit earlier, it's hard to say, we see mammals. Notice that the fossil pattern, fish came first, then amphibians, then reptiles, then birds and mammals. It fits with the molecular evidence, the amino acid sequence or DNA sequence pattern. Here's another example of comparing amino acid sequences. This time we're comparing cytochrome C sequences. Cytochrome C is one of the proteins that make up the electron transport chain in cell respiration. So it's a crucial gene for all organisms essentially to have. We're comparing the cytochrome C amino acid sequences to the human cytochrome C sequence. So again, notice the human and monkey sequences, very similar. Human and pig sequences, a little less similar, but still really similar. Then ducks. Then we've got the reptiles and the fish, the invertebrates, all the way back here to Candida, which is a yeast, so a fungus. So again, notice that the more related things are, the more they're going to share these homologous amino acid sequences. Another example of proof for evolution is artificial selection, or what we might call selective breeding. It's man-made evolution. For example, all the domestic dogs on Earth today were bred to be that way. We selectively bred certain dogs to get the breeds that we have today. Most of the crop plants that we use for food, those have been selectively bred to make bigger, more beautiful, more tasty crops. The original plants that those came from were very different than the ones we use today because we made evolution happen. And the argument is that if humans can make evolution happen artificially, nature can make natural selection or natural evolution occur. We've also had a few examples of direct observation of microevolution. We've been able to see since the 1940s the development of antibiotic resistance in bacteria. We've been able to see since the use of DDT and other pesticides the evolution of resistance, pesticide resistance in certain insects. So those are examples that we can see in the real world. One other thing that I'll briefly mention here are analogous features. These structures don't imply common ancestry. They're structures that are similar in different organisms, but they're not due to evolutionary relatedness. For example, the wing of a mosquito and the wing of a bird. Yes, they're both wings, but the DNA that coded for those wings is very different. Those two wings have very, very different structure. Even though they both have wings, they don't show that those aren't proof for common ancestry or relatedness. So here are some homologous structures. These are forelimbs and vertebrates. These are homologous. These all look the same. They have the same basic bone structure, the same number of bones, because all these organisms share common DNA that codes for these structures. These are homologous features or homologies that show that these organisms are related. Here's the insect wing and the bird wing. Very different structures, made of very different things, coded for by very different genes. These are analogous structures that don't imply relatedness, evolutionary relatedness. All right, that's it. Hope that helps.